cool cats in this episode here. Ooh, sorry, you gotta smile sometimes, right? Actually, gotta smile most of the time. If you're not smiling, what the hell are you doing? What the hell are you doing? Get that smile on. But yeah, where were we? So we're talking about the basic structure of an amino acid. So recall, amino acids are the monomers of a protein, which is a polymer. So amino acids, we're going to look at a picture I drew, a nice pretty picture I drew in a few moments, but there's going to be a central carbon, an alpha carboxyl, an alpha amine, an alpha hydrogen, or just the hydrogen atom, and there's also going to be an R group attached. So let's look at it here. So we got hope y'all could see there. So we got the central carbon, we got the alpha carboxyl, so the C with the double bonded oxygen, and then the hydroxyl attached there. It's called an alpha carboxyl whenever it's in an amino acid because it helps differentiate that says like, okay, this is an amino acid. So this is the structure of every amino acid you'll see. You got the hydrogen atom attached to that central carbon, and then here's that alpha amine. So it's the N with a hydrogen attached and the hydrogen attached. That's the alpha amine. So central carbon, alpha carboxyl, hydrogen atom, alpha amine, and then also every single amino acid will have all these three, the alpha carboxyl, alpha amine, hydrogen, but it also have an R group. And this R group here, as we mentioned before in a prior episode, an R group really can be anything. So the R group in an amino acid is what differentiates all amino acids. So they're all going to have an alpha carbon, all going to have an alpha amine, all going to have that central carbon, which everything's attached to, and then also going to have that hydrogen atom attached to that central carbon. So we got the central carbon, hydrogen spurting from it, the alpha amine, alpha carboxyl, and then this R group, which can be anything, is going to be the differentiating factor between every amino acid. So they all have the alpha amine, alpha carboxyl, central carbon, hydrogen, but the R groups will be the factor that distinguishes one amino acid from another amino acid there. So essential amino acids, moving farther into the discussion on amino acid, essential amino acids. So first off, all amino acids are important. There's a bunch of them, but there's a certain amount of them that can't be converted within the body, so they must be taken through the diet. So that's why they're called essential. So all amino acids are essential, but some need to be actually eaten, ingested through the diet, which allow them to function how they do. So all amino acids are important, but essential amino acids must be taken in through the diet. Essential amino acids must be taken through the diet. Just remember that. It's pretty straightforward there. But so we discussed amino acids are the monomers of proteins, which is the polymer. So a bunch of amino acids bind together to form this larger protein molecule, this polymer. But to start with combining two amino acids, and if you recall, the reason these combine are due to dehydration synthesis here. So the alpha carboxyl and the alpha amine of adjacent amino acids, so two amino acids that are next to each other. So we got an amino acid, another one sitting right there. The alpha carboxyl of the one, and then the alpha amine of the other bind together, remove that water molecule, which allows a peptide bond. The bond between two amino acids is called a peptide bond. So we have an amino acid here, an amino acid here. When two amino acids bind together, it's called a dipeptide. Guess what? When three amino acids bind together, two water molecules are removed, and we have a tripeptide. So it's the same process, we're just multiplying. So the alpha carboxyl and alpha amine of adjacent or next to amino acids bind together, remove water, and bind together there. So we bind this amino acid with this amino acid. That's a peptide bond. That's a covalent bond between amino acids. That's a dipeptide. Now if we add one more amino acid on, remove the water, bind, that's a tripeptide, and guess what? There's one more definition is whenever there's more than 3 amino acids bound together. So whenever there's a peptide bond between each amino acid, but when there's more than 3 bound together, we call it a polypeptide. So dipeptide is when 2 amino acids are bound together via a peptide bond, which is a covalent bond between amino acids. A tripeptide is when 3 amino acids are combined via peptide bonds between each amino acid. That's tripeptide. And then polypeptide is when more than 3 amino acids, so 4 to 20 or 21 amino acids are bound together. All between each one is going to be a peptide bond there. So there's dipeptide, 2 amino acids, tripeptide, 3 amino acids, polypeptides, more than 3 amino acids bound together by peptide bonds here. So we're going to continue our discussion in the next episode with the levels of protein structure. Exciting stuff. We'll see you back here in a jiff. Or Peter Pan, or whatever peanut butter you're a fan of. And I don't discriminate between crunchy or smooth, so let's just get that out of the way first. Love them both.